Services provided by different biological communities can be managed or optimized. Our overall research goals are to, one, determine how different agricultural practices alter biological processes. Two, generate new knowledge on key biological communities that support resilient agricultural systems. And three, provide a new high-throughput quantitative measure to link soil biology processes to the ecosystems services that they provide. I know each year I've come to this conference and this topic is moving up the list as people talk about it. You know, how do we manage this piece that we know has a very important impact on what we're doing? And hopefully we can learn some more here. Join me in welcoming Dr. Laura.
Thank you. Good morning, everyone, and thank you very much for the invitation to come and speak with you today. This isn't an easy topic that I've chosen. When I was asked what I would want to speak about, it was like, well, understanding soil microbiome seems reasonable, but when you actually break it down and try and figure out how we do that, and given the complexity of this system, it's almost an impossible task. I'm basically Sisyphus here, pushing a boulder uphill. But we do have some insights that I'd like to share with you. I think I'm going to move my mic down just a little bit, because that feels... Bear with me here. There, let's try that. How's that? Yep, good. I was just getting a little bit too much feedback on myself there. Anyway, so for the Americans in the audience who may not know where Harrow is, so the Harrow Research Station is a little bit south of Detroit. Whoops, where are we? Is this one not working? It's not very bright, is it? Hang on, I'm just going to check the other one. See if we've got a bit more. Oh yeah, that's better. A little bit more juice there. So the Harrow Research Station is right down there, and Detroit's just a little bit up there. So we're quite far south, obviously. Corn, soybean rotations, primarily corn, soybean, wheat. High humidity area, lots of moisture, fairly long growing season. So all of the research that I'm going to show you today is based on field trials that are run by the different soils researchers down in the Harrow Station. So I can't take any credit for those actual trials themselves. The people that should take credit for them are folks like Craig Drury, Dan Reynolds, and Shuming Yang in particular. So they run cover crop trials and rotation trials and tillage trials and nitrification inhibitor trials and all those sorts of wonderful platforms that I get to go and basically explore and play in. Brent Suraj is working with me in the microbial ecology office, and I do some work with Anne Verhalen as well. In particular, we've been doing some work with the soil undies, and there's a little bit of that in here as well. And Alex Woodley was working with Craig, and he's now North Carolina, South North Carolina, State U, so he's left. So I just want to, microbial ecology research by definition, we're looking at organisms that live in an environment, and to understand what those organisms are doing, we have to understand the environment that they're living in. And without those platforms and those additional expertises, I certainly couldn't do that. So what we're doing down at Harrow is I think, quite unique, probably globally and certainly nationally. We take an integrated approach to looking at those natural capital, the natural capital of agricultural systems. So we have Dan Reynolds working on soil physics, looking at soil structure. Shuming Yang has a large focus on carbon transformations in those systems, and he's moved into cover cropping quite heavily. You all know Craig Drury, of course. He does world-class work on nitrogen management. And hopefully I will be doing world-class work on soil microbial ecology, and I think it's fair to say that we're already doing that. And we're looking at these facets of an agricultural system in a holistic sense. None of these disciplines are essentially silos. We all have to work together to understand how those systems are performing. And if we can get all of those systems lined up and performing appropriately, then we have a sustainable agricultural system. And a sustainable agricultural system is also going to be a productive agricultural system. So it's important to have all of those disciplines lined up. But yeah, sorry guys, it's really all about the microbes. So sorry Craig, sorry Dan. Because when you're looking at things like soil physics, soil structure, carbon transformations, nutrient cycling, pathogen control, it really is that underground biological community that's doing the work. So you have a combination of everything from your larger organisms like earthworms and your large insects in the soil system all the way down to bacteria that are influencing that soil structure. You've got those microinvertebrates, earthworms, and again, fungi and bacteria that are working together to transform carbon. They're working together to cycle those nutrients, whether it's nitrogen or phosphorus or anything else that's in the system. And those communities of organisms working in those spaces, using those resources, are also helping to control pests and pathogens in your systems. So it really is a community effort. And given the complexity of the organisms in that system, trying to parse out which of these individual functions a certain taxa is doing is almost an impossible task. So what we tend to do now is look more towards what is the community doing? How did the community shift under different agricultural practices? And there's not going to be a big, yeehaw, this is the method that we should use to optimize this the optimal level of biodiversity, but I'm hopeful that with the examples that I can give you, we'll come to a better understanding of how what you guys do on the ground affects the life that's living underground. Soils, and some of this is going to be a repeat from those of you who were in my breakout sessions yesterday, but we'll try and keep this fairly brief. Soils contain 25% of our planet's biodiversity. So when you think about that, and you think about everything that lives above ground, it's fairly amazing when you consider that 25% of all of the types of life that we have on the planet actually live in the soil. And we know very, very little about who they are and what they're doing still. So that single teaspoon of soil up there contains millions of different microbial taxa or species and billions of individual organisms. So that's billions of individual organisms in that teaspoon of soil. We mostly still don't know who they are, and to a certain extent, we're not entirely sure what they're doing. They're living their lives down there, they're forming communities, they're competing with each other, they're cooperating with each other, they're cycling nutrients, they're doing all sorts of things. We know it works because we see the end product of it. Higher organic matter, healthy soils, we see residue disappearing, but we don't actually know a lot about how these organisms cooperate to do that. So what I work on are these first 3 rows of these groups of organisms. So the ones that are in the micrometer range, archaea, bacteria, and fungi. So you can't see them with the naked eye. You can see some of them under microscopes. Archaea you can see under really high intensity microscope, like a transmission electron microscope. But for the most part, we haven't been able to do a lot of work with these organisms unless we've been able to culture them. Because we simply, we can't see them under a microscope, and if we can't grow them, we couldn't do anything with them. So in that teaspoon of soil, archaea, we have over a billion archaeal cells are possible in that teaspoon of soil. Over 1 billion bacterial cells are possible in that teaspoon of soil. Over 1 million fungal individual spores or cells are possible in that teaspoon of soil. Your global species estimates for those organisms are great. It's in the millions and it's climbing every year. We're just really getting a handle on the organisms that are present in those systems. Oh, I didn't realize there was a counter there, yay. Because I tend to ramble on, so. Fortunately, in the last 20 years, soil microbiologists have been able to take advantage of some of the molecular advances that have happened in the human health realm. And really, we're piggybacking on that. So sequencing approaches, we've adopted primarily from medical research. And as the price of those sequencing technologies have come down, we've been able to start throwing more and more energy into trying to discover the undiscovered. And I'm seeing this is what happens when you play around with slides at midnight and try and flip them. So this should be there and that should be there. Because we've gone from culture-based not very long ago, and that's how we were able to look at soil microorganisms, to a sequencing-based approach where every arm in this little wheel is essentially the equivalent of an order of organisms. So this would be all canines, for example, and this would be all birds in the mammalian equivalent. So we're able to get a handle on the complexity, but how do we actually make sense out of that? And we've gone from knowing maybe 10% of what was in the soil to sort of thinking that we sort of know about maybe 45%, maybe 40% of what's in the soil. What's really interesting about these molecular approaches where we sequence the organisms that are in the soil is that it's actually enabling us to go back in this direction as well. So we couldn't culture a lot of the organisms that were in the soil because we didn't know what to feed them. It was pretty simple. Organisms don't exist in isolation, and when we're trying to culture organisms in isolation, they may not have all of the metabolic requirements for growth in whatever the media was that we were trying to grow them in, or they may simply not be able to grow on their own. And so there's a lot of really interesting research in the last decades that's come out of some of this sequencing work where we're actually being able to go back and start cultivating some of these organisms that have high potential for both bioremediation, commercial, agricultural use. So it's actually flipping a little bit the other way now in terms of what people are doing. But regardless, those molecular tools are allowing us to actually look into what was considered classically the black box of soil ecosystems, where we knew there was all sorts of wonderful stuff in there, but we didn't know what they were doing. And we're actually starting to be able to open that black box up and investigate some of these organisms that are in there and figure out exactly what are they doing and how are the practices that we're imposing on our land affecting those organisms and the types of functions that they like to perform. Oops, we'll do that. But before we get too excited about sequencing approaches, and we are in the midst of basically what I would term a microbial gold rush at the moment, and you've been hearing about the human microbiome, and you've been hearing all these wonderful things about soil microbiomes and plant microbiomes, it's worth putting it in perspective in a timeline. We're awash in data right now. The sequencing is wonderful in that it allows us to do a lot of really incredible research and uncover a lot of truths, but it's also very labor intensive to work with those data sets. And if you consider that we've known about the use of organic amendments since the 1500s, there's the first recorded, the first records of farmers actually talking about adding organic amendments and the benefits of that to a cropping system are back from the 1500s. We've known about the mineral forms of nitrogen in soil since the 1700s, and research on that has been ongoing essentially for the last 250 years, and we still don't know everything about how mineral nitrogen behaves in soil systems. By contrast, if you look at biological research and microbiological research, by the 1800s, we knew that a lot of these processes here were due to biological activity. We figured that out. In the late 1800s, we figured out that we could grow some of these organisms, and being able to grow them allowed us to study them and figure out what they were doing, get a better handle on that as well. But we were pretty much stuck with these culture-based methods until literally about 20 years ago. And around 20 years ago, we realized that we could extract total community DNA from soil. So you could take basically a ball of soil, we could subject it to some chemical procedures, extract out all of the DNA that was in that soil, and then we could do things with that DNA. And what we've been able to do with that DNA has advanced rapidly over the last 20 years, to the point now where SB2 was actually the soil biology initiative in Australia that I was part of. And so the Soil Biology Initiative was one of the first worldwide, or one of the first large initiatives to harness the potential of molecular sequencing to investigate microbial communities in agricultural systems. So it's really only been 20 years. So managing expectations as to how we're going to be able to interpret that data, I think it's worthwhile having this in context. It doesn't mean that there's not a whole lot of really good people doing a lot of really wonderful work, but we're still really in the early days of what we're doing. So if we get back to the probably most important question for here is, how does what we're doing on farm affect soil life? Well, if you imagine that, again, you've got this complex ecosystem underground, you have organisms with preferred niches, you have organisms with preferred nutrient requirements, anything you do to that soil is going to affect what's below ground. So if you're adding in how you manage your residue, whether you're adding in fertilizer or not, That changes the nutrients available for microbes in the soil system. It changes habitat availability. If you till or don't till, you're altering biopore networks that those microbes might rely on. You have wonderful biofilms within biopore networks. You have 100 times the microbial biomass on the wall of a biopore than you do a centimeter further away from that biopore. So if you till, you destroy those biopores, and what happens after that? Crop rotations, and we've heard a lot about, obviously, crop rotations over the last day. Diversity above ground, we like to say, is equal to diversity below ground, and that's true. But what level of diversity is good? We still don't know about that. If you're adding amendments, manure, and there's a lot of people working with manure, that's going to change your microbial population dynamics. So every little thing that you do above ground that somehow changes the soil environment is going to shift the microbial community dynamics, and it's going to change where your active microorganisms are, it's going to change how quickly they're working, and it's going to change what they're doing. And the question that I've been running up against is, okay, well, that's great, but how long does it actually take? If I do this, will my biological community recover? Is it actually going to recover, and how long will it take to recover? If I implement a new best management practice, if I switch from no-till to till, how long will it take for me to reap those biological rewards? And once I've reaped those biological rewards, is it going to be steady? Or if I have to till 1 year, am I back at square one? Those are really interesting questions, and they're hard to answer because, again, we've had 20 years worth of these tools available to us. And to answer some of those questions, you almost need another 20 to 50 years worth of research. But I've got a bit of a shortcut, which we're going to share with you. So when we talk about microbial communities and soils, I think we have to go back to some pretty basic ecological principles. And I know a lot of people in here would have had some sort of ecological courses in university. So when we're talking about a microbial community, so you've got your microbial community here, it experiences a disturbance, whatever that disturbance is. It might be a rain event, you may have thrown fertilizer on, you may have tilled, you may have changed the type of manure that you're putting on the system. Whatever that disturbance is, the microbial community has to respond to it somehow, because you basically just knocked on their door and said, bleh, here's a bunch of manure. So the microbial community, if it's highly resistant to change, it will stay the same. So if you have a really healthy, robust, usually a very diverse community that has been in place in that system for a long period of time, and that disturbance is something that it encounters periodically, then the community might stay the same, and we call that a resistant community. If the composition of your biological community changes, but after a certain period of time it returns to its original state, we say that's a resilient microbial community. You impose a change, it responds to that change, and then maybe a week later, 2 weeks later, a month later, it goes back to its original state. But often what happens, particularly when you're dealing with changing management practices, is that the composition changes and it remains changed. It doesn't go back to that same original state. And again, if you're implementing new BMPs, you want it to stay changed. But in that case, whether it still performs like the original community or not is very important. And if it performs like the original community, then we talk about functional redundancies. Different organisms in there, but it's doing the same thing, so that's fine. We don't care. And we've heard some about that. We heard some of that yesterday as well. But it doesn't necessarily matter who the members are as long as all those functions are in place, so that's okay too. But you can also get it performing differently. So you impose a change, the community changes, or you impose a disturbance on the system, the community changes, and it remains changed, and it behaves differently. And again, that can either be a good or a bad thing. So if you have a lot of disease pressure in your system and you're trying to alter your management practices to improve pest suppression by that biological community, you don't want to stay up here. You want to actually get down here. And then you want that to become the new steady state. So you want to change it, get it to perform differently, and then flip it back. So it's useful to have these concepts in your head when I go through the next series of slides. And I do sort of carry this little diagram through so we can refer back to it. So how long does that change take? Does it take a week, a year, a day? Does it take 10 years? Does it take 50 years? And when you do succeed in changing that microbial community, are those changes stable? And I'm going to show you some data from some different trials where we can take a look at how 60 years of a management practice has impacted those microbial communities. And we can look at a 10-year chronosequence within that 60 years. We can look at a snapshot after a few decades. So management practice in place for a few decades. Well, how is the community responding over a 3-year chronosequence? We can look at a single season. So you impose a new management practice. What happens at the end of the year? What's the microbial community doing? We can look at 2 weeks. So we'll run through some examples of all of this with this concept of how management is altering microbial communities and what that means for some of the important processes that keep your farm sustainable. I always do that. Okay. Or we can sequence those microbial communities and get a handle on the full suite of organisms that are present in there and start to make inferences about that ecosystem community structure. And we do both. And we do all of this with the background of soil physical data, soil chemical data, yield data, temperature data, moisture data, all of that's important for how those microbial communities are responding. Just doing the molecular work alone would be, quite frankly, really interesting to me, but not to anybody else. So for the first one, we have a management that was imposed about 60 years ago. So the long-term tile runoff study down at Woodsley was established in 1954. Most of these results I'm going to show you are on a Brookston clay loam. This is no different, it's Brookston clay loam, and it's fertilizer and rotation treatments. And what you have are essentially continuous corn, rotation corn, or sod, fertilized or not fertilized, and they've been doing that for 60 years. Now, if you're wondering what continuous corn not fertilized looks like after 60 years, and you haven't had the pleasure of visiting this site yet, these are all taken in June of 2016, these photos. There's your continuous corn not fertilized after 60 years. There's your continuous corn fertilized, so it's still getting its full complement of nitrogen and phosphorus, and yet it's nowhere near coming to this, it's nowhere near as healthy as the rotation corn. So it's an oats, corn, alfalfa rotation. It's nowhere near as healthy as that. So there's something else going on in here that is divorced from nutrients per se. And we know that that's about the soil health itself and other things that those microorganisms are doing for yourself. And even the rotation corn, no fertilized, is not doing very well. In the back, we have a woodlot that has also been uncultivated for 60 years. So we use that for some sort of baseline comparison. So if we're looking at how is this management shifted the microbial communities, it's good to have those baselines to say, all right, here's, if we weren't farming, this is potentially what the community would look like. So when I say that we can use a 10-year chronosequence, we have archived soils at Harrow, and a lot of places around the world have dried soil archives. So the paradigm in my research field has been that you have to sample soils fresh, you have to freeze them, you have to capture that snapshot of the community as it was on that day that the sample was taken. But there's millions, if not billions of dollars worth of soils and the inherent background research that are sitting in soil archives around the planet. And those soils are really, really valuable. So when I got to Harrow and I saw the soil archive there, I started to think, well, can we do anything with this? And when you think when a soil dries down, the organisms die off to a certain extent. Some of the really resistant organisms don't. But even the organisms that die, their DNA remains in the soil. So the DNA really strongly absorbs onto clay particles and organic matter, and that DNA fingerprint is still present in the soil. So I wanted to see whether or not I could extract DNA from some of these older soils that was actually representative of the original microbial community that was there, basically a fossil, if you will, of what was happening at that point in time. So before we could work with that DNA, we figured out how to extract it, so we modified our usual protocols. We're able to pull a healthy amount of DNA out of those soils that have been sitting dried for up to 25 years was the ones that I was working with. Or sorry, 25 years. And we realized that we could extract DNA that was representative of the original environment. So typically what we see when we're extracting DNA from soils is that the more organic matter you have, the more microbial biomass you have, the more DNA you can extract. And we see that pattern. I've seen that pattern everywhere around the planet. That holds true. And as you go up in organic matter from your monoculture corns to your woodlot and your bluegrass treatments, we see this wonderful increase in what I would call molecular microbial biomass. So it just tells me that the DNA that I'm getting still remembers the system that it came from. So that memory, that microbial memory, is still present in the system. And we did some other fine-tuning to make sure that it was reflective of the original environment. So with that in mind, it was, well, now we can look back in time to understand how our microbial communities might be responding to change. So we sort of circumvent, in this case, close to 20 years worth of ongoing research and reuse what we already have and the investments that we already have. So I'm just going to give you a really quick few snapshots of each of these. So this isn't a heavy results section. It's more trying to get some concepts across. So we sequenced those soils. And if I was to throw one of those ordination diagrams up like I was showing yesterday in my breakout groups, and you'll still see a few more of those for those of you who are there, you would see that they're polar distances apart. So communities that are coming from that non-fertilized rotation corn are fundamentally different than the communities that are coming from the woodlot, and they're fundamentally different. They're completely different. It's like you've sampled one of the soils here and you sampled the other one in Western Australia, and you went down to Antarctica and sampled the other one. That's how different those community structures are. But you can start looking in them and pulling out information about the organisms that are present in those communities as well. And I've just given you, again, a quick snapshot here. So if we look only at rotation corn, regardless of fertilization, certain organisms we see starting to dominate. So in that rotation corn, where you're getting varied organic matter inputs going into the system, we see a significant increase in organisms that are able to actually cycle that carbon. So again, from that archive, those archive soils in the DNA, we can pull out organisms that are responding to those management impacts. Similarly, in the fertilized treatments, so again, in those fertilized treatments, whether it was continuous corn or rotation corn, we can actually pull out organisms that we know are associated with disease suppression. So we can start to dive into that sequencing data, pull out organisms for whom we know what the function is, and start to understand how that management over time has affected those communities. And people are currently doing this with things like antimicrobial resistance and some of the herbicide degradation genes as well over time, because you can use these archive soils to capture that period when management changed and then see what happened to the soil community before and after that change. So we can also, when we're talking about concepts of resistance and resilience, we can also say, well, during that 10, 20-year chronosequence, which management practice promoted the most resilient community in my system? And I've just done, this is based on quantitative PCR data, and I've just taken a series from 1994 to 2013. It's not continuous. We don't sample every year sometimes. We're reliant on research funding, and so, people may not sample for a year. But I've taken a chronosequence from 1994 to 2013, and I've pulled out a couple of the more extreme things just to highlight the example. We have continuous corn fertilized and bluegrass systems fertilized. On the y-axis is just gene copies of these particular functions, so gene copies per nanogram of DNA. This is the ability of that soil to mineralize nitrogen under dry conditions. This gene, this enzyme is found in organisms that like stressful conditions. They like it when it's really dry. When everything else, when the system dries out and everybody else goes dormant, the bacilli that harbor these types of enzymes go, yee-haw, it's my time, I can pop up. Bacterial ammonium monoxygenase, you're all familiar with. Those are the organisms, that's contained in organisms that cycle nitrogen from ammonium to nitrate. So we can look at this chronosequence and we can track it with environment events. And in 2005, I'm glad I wasn't here. It looked like a horrible year. It was a drought year. It was a really, really hot and dry year. And if you look at these two systems and you compare your bluegrass versus your corn, what you can see is that you do get an increase in this drought-happy organism in bluegrass, but it's not as extreme. So it responds to that environment, it increases in abundance, but it levels off quite quickly, and it never moves away from that baseline very much. It kind of goes, yeah, I'll respond, and then it keeps going like that. In a more stressed system, you see a huge peak in response and then a crash, a peak and a crash, and that's what you see in your continuous corn fertilized. You see those microbial communities, they're not very resistant to change, and they're not very resilient. For things like ammonium monoxygenase, again, you can see really strong responses in those stress systems. So again, this is continuous corn. As soon as Craig implemented a new management practice, those communities, we went, what the heck? And they shifted really, really dramatically. So whatever you throw at this bluegrass system, the community is diverse, it's healthy, it's able to weather the management impacts, it's able to weather impacts. Whereas those in more stress systems are not as resilient. So we can actually track that, and we can go back in time and start tracking that in some of the other systems that we're looking at. And we're looking at some of these functions as indicators of change as well. So we're moving forward and using those as molecular indicators. So if we look at what happens after 20 years, and those of you who were in my breakout session yesterday saw this, so this is a rotation trial, crop rotation, there's monoculture, and up to 4-year rotations with and without red clover in this system, 144 plots. And these plots are fairly small when you think about it. So the whole system is 108 meters long by 244 meters wide. So it's a fairly homogeneous piece of ground that we're starting with. So the microbial communities in there at the beginning would have been very, very consistent and very similar because they received very, very similar inputs, and they live in an area that's fairly homogeneous. So we went in and sampled over 3 years, twice a year for microbial parameters, spring and fall. And obviously in the springtime, you've got very, very different conditions depending on the crop rotation that you have in place. So how is the microbial community responding in the spring to a corn versus a wheat, and compared to the fall where you have very different crops in play? So this comes down to, is the rotation that you've implemented resistant to the change imposed by the new crop coming in, or does it respond? How stable is that community? Is 20 years long enough to develop a microbial community that can weather those changes that you're imposing by putting a new crop into the system and the new fertilizer requirements? Is it weathering that change? Because the one thing that we do know is that the 20 years of rotation treatments have fundamentally altered the soil environment. This may not seem like a huge difference to you if you go from continuous soybean at 3.34% organic matter to soybean in rotation at 3.84%, but that 0.5 difference to a microbial community is kind of the difference between feast and famine, if you will. Similarly to pH, an altered pH from 6.38 to continuous soybean, which is up from soybean in the rotation at 6.0. From a microbial perspective, if you're living underground, that half a log factor increase in pH is going to alter who's there and your ability to perform different functions. So 20 years, we see a fairly sustained shift in certain soil parameters, and we see this leads to a sustained shift in the microbial communities that are present there. All right, we couldn't get away from these. For those of you in my breakout session, you'll know what these are. So this is an ordination diagram, which essentially takes a large data set and compresses it down into something that can be compared visually. So the distance here doesn't necessarily matter. All you need to know is that 2 points that are close together are very, very similar. Points that are far apart are very, very different. So underlying each of these points is about 20 to 30,000 sequencing identities. So 20 to 30,000 organisms underlying that individual point. So the more similar that community is to another community, the closer they will be together on this ordination diagram. So I think the analogy I gave yesterday was, this is your family barbecue, this is all the teenagers up here hanging out, this is your Aunt Edna down here hanging out, and I have a drunk Uncle David who likes to try and mix it up with everybody. But what you can see is that 20 years, you started out with a community that was likely very, very similar to each other. You're not in a very big plot of land here. 20 years and you've imposed a fundamental shift in the types of organisms that are present in that system. The communities in your perennial grasses are completely different than the communities in your continuous wheat. And I should preface this by saying when I say completely different, what I mean is that the composition is different. So the relative abundance of a bacillus versus a pseudomonas has changed. Bacillus and pseudomonas are still there, but it's the relative composition of that community, the makeup of that community that has changed. Your continuous wheat, this is a fungal community. I should have mentioned that. I think it's there, but so this is the fungi that are present in the system. Yesterday I showed the bacteria. Your continuous wheat, the fungal community in that continuous wheat is completely different. Continuous soybean, different continuous corn. When you have a rotation of those three crops together, basically you get a blend of those communities happening. They never go as extreme as the community, so they never, if you have a corn in play, the community doesn't turn into the same community that you find under continuous corn. What you have is a community that remembers all three crops that were in play. You have a community that has a historical memory of 20 years of corn, soybean, wheat. And so it's more of a blended community. Like this community here is more diverse, it's more abundant, and it's more resilient than these communities here. If you were to impose a change on this, you would see a dramatic shift in the first year. So all of these points here represent 3 years. So that you've got 3 different seasons in here with different different weather events, spring and fall, 3 different years, and yet that's the shift that you see. So that community is now highly resistant to change. It's not budging. It's not budging from spring to fall, and it's not budging from year to year in those systems. So when I showed this yesterday as well, that shift in, and this is based on bacterial data here, that fundamental shift in community structure leads to alterations in function. So you've gone from here, you've gone from your original steady state before the trial started, you've altered the composition and it's now performing differently. It's not functionally redundant from the start. What's happened now is that composition remains altered, it's performed differently, and in all of those systems after 20 years, you're back up here again. And so whatever you're doing from now on, you sort of have to look, well, does it stay the same as a continuous corn or a rotation corn? So you've altered the steady state, it's performing differently, and in some cases, we can track that through functional data. So for example, in your continuous soybean, it is much less diverse, the communities are less diverse, they're less abundant, and you're starting to see an increase in certain specialized organisms. Organisms that perform nitrification, for example, are much more abundant within that community than other organisms like those that cycle organic matter. So you can see that fundamental shift simply based on whether or not you choose to rotate your crop. Long-term impacts. Okay, we're moving down now in time. So we know that 60 years makes a difference, and we can see that 50 years on in a 10-year chronosequence. We know that 20 years makes a difference, and we can see that over a 3-year chronosequence. Well, how long does it actually take to get to that new steady state? And I think that's the question that's of most interest. And so Craig also has, because Craig has all of the trials, Craig also has a Stover trial where they're looking at Stover biomass harvesting originally it was for bioethanol production. So they have Stover removal rates of 0 to 100%. 100% fell off for some reason. And this is tilled or no tilled. And you guys are familiar with this. I mean, 100% Stover removal rate, and we removed this by hand. So it's a little cleaner than you'd see on the farm, but you have a fundamental difference in soil cover there. And when you alter that soil cover, you're altering microbial habitat. So your surface microclimate is different. It's either warmer or wetter or cooler or drier. You've altered the distribution of nutrients down through the soil profile, so like that water extractable organic carbon and particulate organic carbon that microbes really like, where the distribution of that is shifted. And you can see in the first year, you can see decreased CO2, but increased N2O, so increased greenhouse gas production as you remove that Stover. And But you can see the results of the microbial activities, but you can't see anything else just yet after the first year until you look at the microbial communities. These guys are the canary in the coal mine for change. So I'm only showing you 0% stover removal, 50% stover removal, and 100% stover removal. These are bacterial populations. We have the ordination again. The closer two dots are together, the more similar they are. And what you should see is that as you remove that stover, the communities are becoming more different from each other. So you're changing, we have a change in that community structure from a steady state here that it started at, and 6 months later, after a single year of removing or leaving that stover on, you're starting to already change that community. We don't know what the new steady state is going to look like. A lot of this data is really quite fresh. So for example, we do have a third year of sequencing on this, but we're still just parsing for the second year of sequencing. So it'll be interesting to see in that 100% removal, does it actually go back to the original state? Is it resistant to change? It's a blip. 6 months, it's changed, now it's going to go back to the original state. Or has that community, and hence the functions, been fundamentally altered, and you're reaching a new steady state? So we can see as well in that type of data, we can see it in the communities that are present, and we can see it in the functions that they're performing. Again, Craig sees it in, for example, the greenhouse gas emissions, and I see it in the microbial communities that produce those greenhouse gases. So if we look at your classic nitrogen cycle, if we're looking at the cycle as a biological system, it's all linked together. You've got different organisms that perform this step, different organisms that perform that step, different organisms that perform that step, and so on, but they all work together. So they pass, it's like a football team, they're passing products from one group to the other, and if you remove some players or you alter the conditions for one player, you change how the game is going to play out. And so if we look at, again, those functional communities, so if you look at, I didn't do this very well. So if you look at, this is your organic carbon cycling over here, this is your organic nitrogen cycling here, your mineralization, and this is your mineral nitrogen processes. What you do when you remove that carbon from the system is you shift the system towards a system that's really happy to cycle mineral nitrogen, but all of a sudden has stopped appropriately cycling carbon because you've removed a large pulse of that carbon. So you've altered the organic matter input, you've altered the climate, you've shifted how these microbes play with each other, how they function together as a community. And when you do that, you alter outcomes. So you alter outcomes in terms of the amount of nitrogen that's kept within your system. So we see that a single season is enough to shift those microbial communities, both in terms of who's there and what they're doing. So right now in this whole scheme of things, we're here. We're at an altered microbial community composition. We don't know if it's going to go back to the original composition. If it stays this way, we don't know whether or not things will eventually cycle back and everybody will play together and function and cycle through the carbon and nitrogen well together or not. I suspect not. I suspect that what will happen with long-term carbon removal based on what we saw in the long-term trials is that you'll get a system that performs differently. Oops, I'm trying to move on. I think my clicker stopped working. Oh, there we go. All right, I did pop in at 2 months here. So we have what happens after, and this is just a bit of fun, really. So this is part of the soil urundis. 2 months, what happens after 2 months? After the addition of a high carbon but low nitrogen substrate. So if you add a high carbon, low nitrogen substrate into your system, in this case the underwear, and you excavate those underwear, you can actually look at the communities that are present, colonizing the underwear itself, on the underwear soil really, really close to the underwear, or in the bulk soil. So you can actually look and see in 2 months, has adding that carbon substrate fundamentally changed what happens in the community around it, or is it a short shift that happens in the system? And what you can see is that the bulk soil community, so the greater community around that one single carbon addition, hasn't changed that much. Your bulk soil community, again, the closer things are together, the more similar they are still fairly similar. There's not a lot of distribution there. Your undesphere community, we're starting to see a little bit more spread. So communities that are closer to that carbon are responding to that nutrient source. And then the communities that are colonizing the underwear are, quite frankly, all over the shop. So depending on the state of decomposition of the underwear, you actually have completely different communities colonizing that underwear. So in 2 months, you've fundamentally shifted the communities that are breaking down that carbon. That's a given. But we suspect, based on what we're seeing here, is that they'll shift back to the original steady state if you were to leave that carbon in the sun. And that makes sense, right? You've thrown in some carbon. Guys that like carbon have come along and gone, I can break this down, I can work with this. They'll decompose it. Once that material is gone, they'll move to another carbon source. So they'll decrease in abundance there and move to another carbon source. So let's go here. I'm having issues with my clicker. So you see the temporary shift in microbial communities that persists probably until about this stage. At this stage on those underwear, you've gone from this community to this community. So you've seen a complete shift in the community that's present on that underwear, but we suspect, and I think I had a little discussion with Bart about that yesterday, this guy here is a proteobacter that's really, really good at breaking down really difficult carbon compounds, and that's the main organism that's present on these elastics now, and I think it's just getting at the little dregs of carbon that's there. And you can actually track that temporary shift in functional communities as well through quantitative PCR. So there's not a huge I should explain this. So 1 means lots of decomposition. This is a 1. 5 means not very much decomposition. This guy's rated at about a 3 over here. So this is your underwear that's pretty much completely intact. This is the underwear that's completely decomposed. There's not a huge difference in the functional potential of those different soils to actually degrade carbon. So if you look at the cellulose decomposition potential of those soils, it's all fairly similar. The organisms are there. If you look at what's colonizing the underwear itself, if the underwear has already been broken down, those functions are heading back down to a steady state that might be found in the bulk soil. Whereas those that are still intact, you have organisms still on those underwear that are busy attacking that carbon. The one thing that we saw so far that stood out in terms of why some underwear might decompose versus the other, and I don't have management information on that, is based on nitrogen status in the system. So complex nitrogen status, we think, is playing a role in whether or not those communities, those underwear actually decompose. I'm trying to click, there we go. All right, last set. I realize I'm running out of time too. So we can also look and see, all right, does the community shift after 2 weeks? And what does a management practice do after 2 weeks? So from a microbial perspective, if you're adding a urease or a nitrification inhibitor, how long does that actually impact the organisms that cycle urea or nitrogen? And again, this is another one of Craig's trials, where he's looking at different fertilizer formulations and different application methods. We can go in and have a look at how the different formulations impact, in particular, this nitrification step of the cycle. So this is sort of the step that you're trying to stop with a nitrification inhibitor. And what happens to that organism that's performing that step? Does it recover quite quickly once the nitrification inhibitor is applied, or does it stay suppressed? I have a lag here with my, there we go. So we're going to look at quantitative PCR data here. So this data represents that nitrification organism. So it's your bacterial nitrifiers in that system, and it is the target for the nitrification inhibitor. On the bottom, what I have is the abundance of that organism in the soil system. On the top, I have the activity. So this is DNA-based versus RNA-based. So I look at this as the difference between, I have 20 teenagers living in my house. They all have the functional capacity to run a vacuum cleaner. So that's their functional capacity to vacuum the floor. Up here is the actual potential that they're vacuuming at any given point in time. And I would say most households, it's probably down around there. So that's the difference. We know these organisms are present. And what we can see, if you look over time, so you have your control, your urea plus urease inhibitor, and your super U on the bottom here, from 1 day to 21 days after application, rather. That planting is wrong, after application. And you can see that, the abundance of those organisms isn't changing in response to those nitrification inhibitors. You'd have to squint really hard to see a difference between these. So this is just, again, so gene copies of that function per unit of the microbial community present in the system. So that community is resistant to the application of that inhibitor. It does respond to it, but it responds to it in a very short time frame. So if you look at your urea here, your nitrification, the activity goes up quite quickly. Within the week, you're actually at a maximum nitrification activity after you've applied the urea. If you add an inhibitor in, you suppress the activity for an additional week. So basically, the organisms are still there, but they're not functionally active until about 2 weeks in. After 2 weeks, they're as active as they're going to be. So somewhere between that 10-day period and the 14-day period, the efficacy of that inhibitor wore off, and the microbial communities basically woke up and said, I can vacuum the floor now. So in this case, there is an immediate impact, but the communities, in this case, specifically the nitrification community, is resistant to change. So it doesn't change. The abundance of those organisms present doesn't change. The activity alters, but it goes back to normal really, really quickly. So it's both resistant and resilient from a microbial perspective. So this is a minor disturbance for bugs. I'm trying to. Yeah, I'll go there. Minor disturbance. So if we're looking at how agriculture affects soil life, and I don't want the cowbells, I am almost done, just 2 slides. Well, it depends, right? It depends on what the disturbance is. Altering carbon inputs into the soil is a big disturbance from a microbial perspective. Adding in something like a nitrification inhibitor is a minor disturbance from a microbial perspective. So it's the type of disturbance and the duration of the disturbance that actually matters. And what you need to worry about from a microbial perspective is the endpoint that you want to reach. Are you already at a healthy state? Then you want to stay up here. You want to stay in this realm here. If you're starting from a degraded system, you want to eventually reach an altered composition and have a community that functions better for you, has higher disease suppression, cycles carbon better, cycles nitrogen better. So there's different ways that we can look at it. So I'm going to leave with something like this. I have one more after this, but it's a complex living system. What you do to that system, whatever you do to that system, is going to shift something. It's going to alter something. There's a finite niche space, finite habitat in the soil. Soil is not infinite from a microbial perspective. They have competitors, they have collaborators, they have to be able to be in close access with those organisms. They compete with each other for niche space, for nutrients, for all of that. And so anytime you change that soil, you're going to change how these communities interact with each other, with flow-on implications for what those communities are doing for you. So if you're hoping for a simple answer, there's no simple answer, because there aren't. I have one last slide. So agriculture activities are going to impact your soils at different levels of scale, so different spatial scales and different time scales. So we can detect some of those. I mean, some of the advances in soil microbiology means that we can look backwards in time now and see, all right, how have things changed over up to half a century? And the oldest paper I've seen has been actually about 100 years, where they've looked back to see 100 years ago, what was the soil community like? And we can use the same techniques to essentially say, all right, what happens 2 weeks from now? And in each of those time frames, we can detect shifts in the communities. And those shifts are entirely dependent on what you've done to the soil. You have complete control over that because biology responds to management, and I think that's a good take-home message. So those responses that you're looking for start as soon as you impose that new management practice. How long it will take for those responses to become stable is anyone's guess. We're figuring it out. Sometime longer than a year and less than 20 years is about all I can tell you. And whether that changes for the better or for the worse is really up to you and the management practices that you use. And I'll leave it at that. Thank you very much for your time. right to the wire. Oh, but-
Excellent time management here. Understanding that, I guess a couple of things I'd like to understand is, so you give us the example, the good example for a lot of us with children. You know, they may be present, but they're not doing what they're capable of doing. If we look at microbes in our soil and, you know, we discover that this is beneficial, that is beneficial, what's going to be our pathway to introducing those microbes to have that benefit?
Yeah, that's an excellent question, and that's another active area of research. And I think it comes down to, I was asked the question yesterday by somebody about, well, this is lovely information, and the information itself is useful, but how do we apply it? And I think that's the same. If we can understand, so if I can find that magic organism, if I can say that the organism that is stimulating the majority of the underwear decomposition is a particular strain of Sphingomonas, and it very well might be based on what I've seen. That's the main organism colonizing those underwear. And we want to promote that, then we can look back and say, all right, what are the requirements? What are the habitat requirements of that organism? So if we want to promote a particular type of organism, if we understand what its needs are, then we can shift the system either temporarily or permanently to favor, to promote conditions that favor that organism. So that's one way. So that's altering management practices. And you guys are all doing that from what I've seen. You're all doing that anyway. The other thing is there's a lot of ongoing research on inoculants. And that's where the molecular world is helping, because most of the inoculant formulations that are coming out now are no longer single species. They're complexes of organisms, because we now know that single species don't do very well by themselves. So you can inoculate, and there's work on that.
Okay. A couple of questions from the audience here. First one is concerning soil amendments. Is there potential for concern when a biological soil amendment is applied and microbial populations are affected, either positively or potentially negatively?
Yeah, okay. So when we're talking biosolids or compost? I guess before I start answering that question, remember that the soil's already has a population that's already there. When you're adding biosolids, for example, any organisms that are present in that manure, in that compost, in those biosolids are going to have to compete eventually with the organisms that are already present in your soil. If you have a diverse community in that system, the chances of any one single organism taking over is from your biosolid or manure application is very, very low. Having said that, one of the things that a lot of people are concerned about and are worrying about are human pathogens, in particular if you're using biosolids, because you can actually track the introduction of antimicrobial resistance genes into the soil reservoir after the application of manures and biosolids. So it's a bit of a yes, you should be concerned. No, it shouldn't stop you from applying manures or biosolids to your system, because the benefits outweigh the negatives, and let the rest of us worry about how we're going to make sure that the manure and the biosolids you get don't have those pathogens in them to begin with. And the speaker, we saw a really good example of the weed seeds coming out of the non-composted manure this morning. So there's things that you can do if you're concerned about the source of your biosolids to minimize the risk of any potential pathogens in your system.
Okay. And this next one kind of falls on the line maybe of unintended consequences, but have you done any research with the change in active communities with or after herbicide, insecticide applications?
Yeah.
Or even fungicide is supposed to be right down the line too.
Okay, the short answer to that is no. Sorry, I haven't. So there is some research out there on that. Again, the majority of your herbicides, pesticides, fungicides they're organic compounds. Microbes like organic compounds. The reason these things are marketed to you is primarily because we can show that they break down in the soil environment, that microbes are able to process them. Having said that, there certainly have been studies, there's supporting evidence to show that some of those compounds actually, the microbes aren't completely effective at breaking them down, and they can be translocated into water systems. There's also evidence to show that some compounds might inhibit some symbiotic relationships that you might want to have in your system. So for example, If you think about things like mycorrhizal colonization of your roots, so those beneficial fungi that colonize your roots and act as de facto extensions of your plant roots, you have a very short window for those organisms to enter into that symbiotic relationship with your plant roots. Similarly for rhizobia, very, very short window for everything to line up appropriately such that those organisms are able to form that symbiotic relationship. So there's evidence to show that some of the chemicals, I'm not going to name names, that we apply to the system might disrupt the ability of that symbiosis to actually take place. So when you're applying herbicides and pesticides, I think it's worth considering what else is happening underground at that stage and whether or not you might be interrupting a courtship that you might not want to interrupt at that period in time. I think might be the best way to put it.
Okay, thanks. And a final question here. It starts prefacing with a statement here, great research, but the focus needs to be on the future, which is no-till with covers. We need to move on from the tillage. But their question around this is, who do we talk to change this?
Yeah, okay. So this is a snapshot of the research that we do. We also, I should mention that we also look at diversified landscapes. So we're looking into the benefits of keeping buffer systems in play around the field, adding in small wildlife habitats into the field. We work on cover crop systems, on intercrop systems, and we're doing similar biological research on those systems. Organic systems down in Harrow, Shuman Yang has some of those. So we are actually doing that work. I didn't show it here mostly because it's not as advanced from my end of things, but it's certainly well advanced from the government perspective in terms of research. And I know I can see people sitting in the audience today that are working on, some of the field trials at Elora or Ridgetown. So, I mean, there is a lot of that going on. The shift is towards no-till and cover crops, and I think you guys are the people that need to be talking to people, not necessarily me. And I think you guys taking that message out and convincing those sort of late early adopters and the rest of the hump to follow along is what needs to happen, because they're not going to listen to me, let's face it. I'm a talking head up here, but they will listen to you guys.
Okay. Thank you. Thanks for a great presentation and taking the time to answer the questions. Thank you.